It will be.
Okay.
Yes.
Awesome.
Yeah.
All right. Thank you so much.
You're welcome. All right, you guys. 12:50. Let's go ahead and get started. Okay, so one announcement. We're going to start the GI Physiology today. I think a lot of nutrition students probably have been waiting for GI Physiology. I think you'll really appreciate some of the things we're going to be talking about today. But one thing that I want to tell you guys right up front, there are a lot of slides and information that are not in your textbook. So it's really important that you guys come to class. I'm going to explain all of the diagrams that don't have text associated with them in detail in class. Now, I know some of you guys do have school-sponsored trips that you're going to have to take. So I am going to try to record. I can't promise anything, but I did actually turn it on today. I'm going to try to record the next 3 lectures so that you guys have some reference to study from for the final. Remember, the final only covers GI physiology and endocrinology. It's not a cumulative final, okay? All right. So remember, if I haven't said this before too, I want you guys to open up your e-poster site on Friday, just to let me see your progress. It doesn't even have to be close to being done, so don't worry about that. You're not getting any kind of score associated with it. I just want to check to see that everybody's kind of starting to put their PowerPoint presentation together. Remember, it's due 2 weeks from Friday. 2 weeks, the very last day of class. All right? All right, so let's go ahead and get started. Let's see here. All right, so I particularly like GI physiology because it is a true organ system. It is an organ system because it's made up of a lot of different organs that all play a role in digestion of food and absorption of nutrients. Okay, really important to understand. We're going to be talking about a lot of different organs with the GI tract. Okay, so starting off, just some general information. Digestion of food and absorption of nutrients are accomplished in a long tube connected to the external world at both ends. This is actually what's called the alimentary canal. Alimentary canal. So I'm going to write that on the board here. Basically what that means is if I took a tube and I put it in my mouth, I wound it down my esophagus, my stomach, my small intestines, large intestines, and let it come out my anus, that would be the alimentary canal. All right, so like this actually says, it's connected to the external world at both ends. And again, like we talked about with the kidney, we're also going to be talking about secretion and absorption. So secretion, if you guys remember, is when you move solute or molecules from the circulatory system into the GI tract. That's secretory, that's secretion. If you're moving molecules from inside the GI tract and absorbing them into your body, remember that's called absorption. All right, secretion and absorption. We're going to be talking about that as well. All right, so that leads us to this next slide. Like the kidney again, we're going to go through 4 main processes. Okay, so you got to know these 4 major processes in the GI tract. It includes digestion, secretion, absorption, and motility. So what is motility? Motility is coordination between nerves and the GI tract to help food keep moving down the line. Okay, so it's also has a lot to do with secretion and absorption, but essentially your GI tract is one of the most innervated organs in your entire body. So motility is a big part of that. Okay, motility is a huge part of that. Okay, so some of the organs that we're actually going to be discussing. Today we'll concentrate on the upper GI tract. We're going to be talking about the teeth, the mouth, taste receptors a little bit, and we'll probably get to the esophagus and what's called peristalsis. On Friday, we'll be talking about the stomach and the small intestines, how the pancreas plays a role in secreting enzymes and bicarbonate to help with digestion. And on Monday, we'll get to the lower GI tract and talk about the defecation response. Okay, so as you may already have guessed, the GI tract is not glamorous. It's not a glamorous organ. We'll be talking about vomiting, regurgitation, diarrhea, and the defecation response, okay? All fun stuff. I really actually like to talk about that stuff. It's pretty fun. So that's where we're going with all that. Many functions in the gut are found in specific locations along its length. Most of the absorption of nutrients actually do happen in the small intestines. So that's really important to understand. We're going to be talking about secretion of acid in the stomach to help chemically digest. The stomach is actually a pretty violent place too. There's a lot of torsion that's associated with it. There's shortening of the stomach, constriction of the stomach, and actually oblique muscles like literally churn the stomach to help with mechanical digestion and chemical digestion with the acid that's secreted. In the small intestines, you also have the pancreas that's delivering enzymes to further help digest the food. So that's where we're going with all that. Now, with this, just an overall general information too, is how is water or fluid secretion and absorption in the gut. This, to me, is astounding when you think about it. On a day-to-day basis, look at the numbers here. You have about 1.5 liters of salivary secretions that occur. 2 liters of gastric secretions in the stomach. You have almost 7 liters of fluid that is absorbed in the small intestines. I mean, that is a lot of fluid if you think about it on a day-to-day basis. And then 1.4 liters in the colon. You actually only, I shouldn't say the word poop out. Let's see how it's, 100 mils of fluid are excreted in the feces, right? So you really don't get rid of a lot of water by excreting it in your feces, right? It's usually secreted and absorbed in the gut, right? In basically these tubes. So think about this too. When we get to the small intestines, we're going to talk a little bit about cholera toxin. Maybe some of you guys have heard of cholera toxin in the news when there was like an earthquake in Haiti, and weeks and months after that, a lot of people suffered from cholera. Exactly what that is, it actually is a toxin that binds to a GS-coupled receptor that keeps the epithelial cells in this on position. So they, instead of absorption of fluid in the small intestines, it's a constant secretion of fluid. So imagine that. You can literally lose your entire bodily fluids in a 24-hour period. That's how devastating cholera can be. Okay, so we'll talk about the mechanism of cholera toxin and how cystic fibrosis, patients with cystic fibrosis have an advantage when they contract cholera because they don't have that chloride channel, they don't have as much secretion, they don't lose as much fluid, and they usually tend to survive a cholera outbreak, which is interesting. Okay, so that's where we're going with this too. We'll talk about that on Friday. Now, I want to also make sure that you guys understand that each one of these kind of these places in the GI tract, they're all considered different compartments. okay? And each compartment has a very different environment. You can imagine with the stomach, after you eat a meal, we've talked about this a number of times before, you literally secrete hydrochloric acid. So imagine you have to keep that acid in the stomach. It can't actually bubble up into the esophagus or get delivered into the duodenal space, which is the small intestines. It can be disastrous, right? You guys know what happens when acid gets into your esophagus. A lot of people know that as heartburn. We'll talk about that lower esophageal sphincter that helps to keep the acid in the stomach. If acid gets into the small intestines and it's not neutralized, it can cause ulcers, duodenal ulcers, okay? So these compartments are important, and keeping the environment within the compartment is also really important. So we'll be talking about sphincters. So obviously this slide is a lead-in to that. There are specialized compartments that increase the efficiency of digestion. And there are muscular valves called sphincters that control the passage of food from one compartment to the other. Sphincters, or these muscular valves, are made up of circular smooth muscle. Circular smooth muscle that help, they have a basal tone to them that make sure that the compartments can contain their environment. Compartments can vary in pH, enzyme composition, and types of secretive and absorptive cells. So again, they have their own cell types, they have their own environments. Again, this happens to maximize efficiency of digestion, all right? Now, this is also interesting too. There's a complexity of gut morphology. There's some real differences in the GI tract when we look at a lot of different species. So I just wanted to highlight, since you guys know a lot about osmoregulation, again, we just finished renal physiology. You guys have some knowledge of this. I just wanted to highlight briefly freshwater fish from saltwater fish, okay? And I'm going to just concentrate on the GI tract, which is right here, of salt or marine fish. Now, freshwater fish live in fresh water. They live in a very hypoosmotic environment, right? So they're constantly, passively taking on water, right? There's an osmotic gradient for the movement of water to go into the fish, and they're constantly losing salt to their environment. So they have mechanisms basically in their kidney system and in their gills to absorb or transport salt into their body to combat that passive loss of salts. Marine fish are a little bit different. It's like living in a desert. They're constantly losing water to their environment and taking on salts because they live in a hyperosmotic environment. Okay, so that's their issue. I think it's interesting that the marine fish actually have to drink water, right? Freshwater fish don't have to do that. Marine fish actually have to ingest seawater. And then you can actually see, here's their gut right here. Think about their gut like the thick ascending limb. Okay, you guys know something about the thick ascending limb. They have the sodium, potassium, 2-chloride cotransporter. So they're constantly absorbing salt, and unlike the thick ascending limb, water follows. Remember, in our kidneys, water doesn't follow. But that's a way for them to ingest salt and take up water. They don't have any other mechanism to do that. They don't have a loop of Henle, so they can't concentrate urine. But this is how they take up water. It's all in the gut. And then they actively get rid of salt through their gills. Okay? But that's how they take up water, which is kind of interesting, I think. And that all occurs right here in the intestines. So you can see there's some real differences when you think about the morphology, gut morphology, across different species. Okay, but what we're going to be concentrating on today is basically the upper GI tract here. I don't know if you guys know of this national meeting called Experimental Biology, or EP. Sometimes undergraduates are invited to go. Physiology is always very well represented at this national meeting. It's in Chicago this year, this weekend. And I was there a few years ago, and I was attending a refresher course on GI physiology. And there was an elderly gentleman there, a scientist that had been in the field for many, many years. And he went on this rant, this diatribe, literally just went off on a tangent, and talked about how the teeth are a big part of the GI tract, and it's totally underrepresented. So for that, since I've been to that talk, I always kind of make sure I talk about the teeth. I have had animal science students actually go to dental school, so I just think it's, you know, a good place to start. So let's talk about the teeth first. The teeth are very important in a process, and I'm sure you guys have already heard this process called mastication. Okay, mastication is just a super fancy word for chewing. Okay, so this is where the food actually enters into your mouth, and you start to mechanically digest food. All right, so you actually have different types of teeth, molars and premolars. They kind of flatten. They are for chewing and grinding. Okay, so you have 12 molars and 8 premolars, chewing and grinding. Then you have humans have 4 canines and 8 incisors, and those teeth are for biting and tearing. That's for a lot of carnivores out there. So we have 4 canines and 8 incisors. Okay, so here's where we're starting to mechanically digest food. I'm just going to briefly go through the anatomy of the tooth. I think there's some really interesting facts about teeth in general. The enamel here, this is the hardest substance in the human body. The hardest substance. 96% of enamel is made up of crystalline calcium phosphate. Okay, so again, the enamel is one of the hardest substances in the human body. All right, dentin, right underneath that, is calcified tissue, dentin. And then in the middle here, you see the pulp cavity. That's where all the living tissue is, in the pulp cavity. So you have living connective tissue, you have blood vessels, you actually have lymphatic vessels. The lymph system actually goes all the way up into the pulp cavity. And you have nerves. The nerves run from the tip of the tooth here, of the root, through the root canal, into the pulp cavity, okay? So the root canal is where a lot of those vessels, nerves, and lymphatic vessels lie. All right, so then you also have the periodontal membrane. The periodontal membrane actually has what's called cementum. Cementum. So, before I get too far down this line, there are 4 major parts of the tooth. Enamel, dentin, the pulp, and then the last one is cement. Okay, so cementum is actually elements within the periodontal membrane that help make sure that the dentin and the bone, that the tooth stays in place, right? It's basically making sure, like the name suggests, cementum. It helps keep the tooth in place. That root canal stays right in place and connects with the bone. And then finally, the gingiva is the gum, right? So everything above the gum line is called the crown, and everything below the gum line is called the root. And that's as far as we're going to go with teeth. But I still think it's an important thing to know. Again, this is all part of chewing and mastication. So let's go ahead and move on to salivary glands. All right? Big part of eating as well. There are 3 salivary glands that I want you guys to know. The first one is the parotid. Second one is submandibular gland. And the third one is the sublingual gland. All right. Now, salivary fluid is usually made up of mucins, that mucus, salivary amylase, which is an enzyme that starts to break down carbohydrates, polysaccharides immediately. This is salivary amylase. Bicarbonate, that neutralizes any acid in your mouth. And lysozyme, which is an antimicrobial agent. Okay? So let's take a look at these different glands. Let's start off with the parotid gland. The parotid gland is the largest of all the glands. You can actually see this. It's up towards the upper jawline. Now, this is weird, but if you actually take your tongue and you kind of move it up towards your upper part of the cheek, you might kind of detect a little bulb. That is where the parotid gland actually secretes the saliva into your mouth. Right about here. Right? I can feel mine. I don't know. Okay, so that's the parotid gland. Now, at rest, when you're not eating, it makes up about 25% of all the salivary fluid that is secreted at rest. When you eat something, it ramps up to about 50% of salivary fluid. All right, the submandibular gland, you can actually see is the next largest. Yeah?
Did you say 15 or 50?
50. Yeah, it goes from 25% at rest to 50% when you're eating something. Oh, let me mention something too. The parotid gland actually secretes amylase and a very serous fluid. Serous is like serum-like or plasma-like, so it's very watery. Okay? It's a very watery fluid that contains amylase. Now, submandibular gland is down by your lower jaw, and it's the next largest. At rest, it represents about 67% of salivary fluid. 67 to 70%, yeah, of salivary fluid. And it's a mixture of serous or watery fluid and mucus, mucins. It's a mixture. And then the smallest gland, it actually only represents about 3 to 5% of salivary fluid at rest. That sublingual gland right underneath your tongue, it secretes a lot of mucus. Lots and lots of mucus. I mean, 3 to 5% of the total salivary fluid, but it is almost all mucins. So I always think about my dog. You guys, I know, have seen dogs do this, but my dog is super polite. I do. I'm not how to say that, but when I eat dinner, he just stands there and stares. He's a yellow lab. And then all of a sudden, he's got these long, mucin-like secretions that will literally touch the floor if I let it. Right? They just are like super stringy. You guys know that, right? I always think about the sublingual gland when I think about that. Almost all mucins. Yeah, so Pavlov's dog. Okay, so just to wrap up, the salivary glands are actually exocrine glands. I don't know if you guys remember a long time ago when we talked about epithelial cells, and I talked about the different types of epithelial cells. One of them was an acinar gland. It has a bulb portion, that's an acinous portion, and then a long duct. So the salivary gland is much like this. I'll flip to the next slide and I'll come back. So you can actually see that it has an acinous portion, a bulb-like portion, and then a long duct where the fluid is actually modified. Okay? So that's known as an exocrine gland. because the secretions are delivered to the outside, and what I'm talking about is into the mouth, okay? Again, why is saliva important? Saliva is important because it lubricates food. The parotid glands secrete amylase, and that already starts to break down carbohydrates like polysaccharides. It helps to start dissolving food so that you can actually trigger those gustatory receptors or taste receptors. And then again, it secretes lysozyme. So you'll already start to cleanse the mouth with antimicrobial properties. So, one final slide on salivary glands. Let's take a look at their anatomy and exactly what's happening. The diagram, like I said, is a little tough to understand if you're not in class. Okay, so these little structures here they're basically capillary beds. Okay, so this is the blood supply here. And these are neurons that are innervating the salivary glands. I think what's cool about salivary glands is they're one of the only structures in your body that are both stimulated by the parasympathetic and the sympathetic nervous system. So if you get an increase in tone in either one of those, you get an increase in salivary secretion. So the fluid is transported first into the acinus. These are the primary secretions. And this is always a good test question, you guys. The primary secretions are isosmotic. That means they have the same osmolarity as blood, about 310 milliosmoles. So it's very plasma-like. Isosmotic. Isosmotic? Oh, okay. So iso means same, and then osmotic. Yeah, isosmotic. Isosmotic. Yeah. So it's I-S-O-O. No, it's actually I-S-O-M-O-S. I'm going to have to write it. I can't even do it in my head. Okay. You could do isosmotic, or sometimes it's written isosmotic. So remember, this means same. Oops. And that's referring to the water transport. Yep, isosmotic. Okay, so the primary secretions are isosmotic, about 310 milliosmoles. The fluid enters into the intercalated duct first. You can actually see that there are these myoepithelial cells that can constrict, so they can regulate flow into the striated duct. They can if they constrict, they're going to increase the resistance and decrease flow, right? Or they can be relaxed, allowing for more fluid to flow into the striated duct. Now, one thing about these myoepithelial cells is that they're also progenitor cells. You guys know that? Stem cell-like. So they replenish the epithelial cells in this particular area, okay? And then the striated duct is where the fluid is actually modified. So sodium and chloride are reabsorbed into the body, whereas bicarbonate and potassium are actually secreted. So again, bicarbonate is secreted into the mouth to help neutralize acid in your food. All right, pretty good with that, you guys? Any questions so far about salivary glands? All right, so let's move on to the taste receptors. I don't really have a whole lot to say about taste receptors, but I do have a few things. You guys already know that there's different taste receptors. There's 5 established taste receptors. We've got sweet, salty, sour, bitter, and one called umami. You guys know umami? Yeah, umami. All right, so umami is umami. You didn't know how to spell that. Umami is a receptor that detects amino acids. So they will detect things easily, like monosodium glutamate, like that's MSG. Other ones are, it gives you, if you guys do any cooking, you might know that savory taste. So this is the savory taste. It basically is in anchovies, mushrooms, right? Sometimes in the cookbook it might say, add the umami bomb, right? I add umami bombs all the time. Okay, so those are amino acid rich foods that trigger these umami taste receptors, that savory taste. So what I was also reading is that there's a new one since September of last year. They actually think that starch has its own taste receptor, right? Starch is a complex carbohydrate, right? But if they block the sweet taste receptors, people can still detect starch. So that's why people love pasta, right? Pasta is a lot of starch. Yeah, right? Yeah, exactly. So this is considered maybe a sixth. There is a paper out from Oregon Health and Sciences, that organization, that it's actually from their Food Science and Nutrition Department, that have shown that this could be a sixth taste receptor, which is kind of cool. Okay, so I do have another thing I wanted to add. There's a paper on your Moodle website, and it actually shows that you have sweet taste receptors in your small intestines. It's not just in your mouth, it's actually in your small intestines. And what happens is when you eat something super sweet, even artificial sugars, it triggers those sweet taste receptors in your small intestines, and you immediately insert more sodium glucose transporters, which allows you to absorb more glucose into your blood. You can really elevate. So what that means is if you're actually drinking a Diet Coke with a lot of pasta, you might think that, right, it wouldn't trigger any sweet taste receptors probably in your mouth, but in your gut, you're going to be able to absorb way more sugar by drinking that Diet Coke than if you were just drinking water. Okay, so what that means is a lot of people think that they might be losing weight by drinking Diet Coke, but in fact they may not be able to lose that weight. It depends on what they're eating with that Diet Coke, okay? So, interesting stuff. The paper is on your Moodle website. It's not required, but it actually is labeled like interesting read, if you guys have noticed that already, okay? So check that out. All right, so now just moving down the line. We're going from the mouth now to the esophagus. Let's talk about deglutition. Deglutition. Everybody know what that means? What does that mean? What's that? It's a very fancy name for swallowing. Fancy name for swallowing, deglutition. So that's where we're going. We're going to talk about swallowing and the swallowing reflex. Okay, so in general, the swallowing reflex is coordinated by the medulla oblongata, the brainstem, which stimulates the appropriate sequence of contraction and relaxation in skeletal muscle, sphincters, and smooth muscle groups. So we're going to be talking about this in pretty much a lot of detail, actually, in just a second. But let's break it down. There's two phases. There's a voluntary phase. You have complete control over it. And then there's an involuntary phase. Okay? Then the swallowing reflex takes over and you can't do anything about it. Okay, so the first part of it is swallowing begins as the tongue pushes the bolus of food. Okay, so what that means is when you chew up your food and you make it into a big wad, I'm going to start calling that a bolus. They call that a bolus of food. okay? So it's you voluntarily take your tongue and you move that bolus of food into the back of your throat, into the pharynx area. All right? Then before swallowing, muscles keep esophagus closed. So you still have the upper esophageal sphincter is pretty tight. Now here's where the reflex takes over. With the reflex, you can see this white area right here is the hard palate. And then there's a soft palate right here. You guys also know the epiglottis. Remember when we talked about respiratory, the epiglottis is an omega-shaped structure that moves over and protects the windpipe, the trachea, when you swallow food. So the involuntary phase is when the soft palate, right next to the hard palate, moves upward to protect the nasal cavities up here. And then number 4 here is when the epiglottis bends to close off the trachea and the lower esophageal sphincter relaxes and opens up. Now that works great. The soft palate again moves up to protect the nasal cavities unless you're like drinking milk. A lot of times it happens on a date, super embarrassing. Maybe you laugh and it comes out your nose. That's when everything goes wrong. But usually the soft palate moves up and protects the nasal cavity, okay? All right, so what's the esophagus all about? I try to make it more exciting. It's pretty boring, but there are some things to the esophagus, okay? So hear me out on this. The esophagus is really a tube that transports the bolus of food from A to B, from your mouth to your stomach, okay? So, but it has some interesting features. It's basically, let me just go to this next one. It's basically made up of 2 sphincters, which I'm going to talk about. And there's a real difference between the upper esophagus and the lower esophagus, okay? So here's a diagram on the very far left-hand side of the esophagus. You do have the upper esophageal sphincter, or UES, that does have a basal tone associated with it, meaning there is some pressure associated with it because those circular smooth muscles are constricted. So there's a basal tone. It's always kind of closed off. Then you have the lower esophageal sphincter. This is the sphincter that basically is the entryway from the esophagus into the stomach. And there's also a basal tone associated with that as well. Now take a look. This is always a good test question too, you guys. Take a look. The upper esophagus is under skeletal muscle control. Skeletal muscle. You would think that would be voluntary. I don't think it's voluntary. I don't think I have any choice in the matter of that one constricting. The lower esophageal sphincter is under smooth muscle control. Okay, so I always wonder, since the upper esophagus is under skeletal muscle control, is if anybody does have control of it. You know those sword swallowers? Or did you guys ever see America's Got Talent, where that guy just swallowed a bunch of stuff and then he could hit his stomach and it would pop back up? I'm wondering if he has any control of the skeletal muscle in the upper esophagus, because it does seem crazy weird. Anyway, my point is to remember that the upper esophagus is under skeletal muscle control. Remember myasthenia gravis? Guys, if you go back to the muscle section, that was when antibodies attack the nicotine-acetylcholine receptors and there's profound muscle weakness. Well, dogs can't come up to you if they have myasthenia gravis and say, I have generalized muscle weakness. Usually they just look super lazy, right? But to diagnose it, they exhibit something called megaesophagus because they have general muscle weakness. Their upper esophagus actually is completely relaxed and it's much larger than the bottom part. They actually get food that accumulates in the upper esophagus because of myasthenia gravis. So if you image a dog that has myasthenia gravis, they're going to have generalized muscle weakness in that upper part. Good way to remember that there's really a distinction between the two sections. Okay, so let's look at this panel right here. That's a little bit to the right. The y-axis here is pressure in millimeters of mercury. The x-axis is in time, okay? So what you're looking at is a bolus of food as it moves through the esophagus in time, all right? So let's just take a look at this top portion right here. You can see that there's no pressure associated with this area when there's no food. But when a bolus of food enters into this area, you can see that the pressure rises, and then as it passes down the tube, it will fall back to its original value, okay? So this is what happens when a bolus of food is right here. At the same time, the bolus of food is right above the upper esophageal sphincter. You can see that there's a relaxation of the upper esophageal sphincter. It has a basal tone. See, it's not completely at 0. It has a basal tone of about 30 millimeters of mercury. And then when the bolus of food is up here, the upper esophageal sphincter relaxes, and then as the bolus of food moves through, the pressure increases and then goes back to 30. Does that make sense, you guys? There is a relaxation of the upper esophageal sphincter when the bolus of food is right here. Now, take a look at this. At the same time, let's go all the way down to the lower esophageal sphincter. There's an anticipatory relaxation of the lower esophageal sphincter before food has even come close to that area, right? That's kind of weird. There's an anticipatory relaxation, okay? So now the bolus of food, you can see, is here. There's an increase in pressure and a decrease. Now it's here, increase in pressure. Now it's here, there's an increase in pressure and decrease. So this is just documenting that bolus of food as it moves down the line. And then now the lower esophageal sphincter is already relaxed and it can slide right into the stomach. So remember, the take-home message here is there's an anticipatory relaxation of the lower esophageal sphincter. That's all coordinated by the medulla oblongata, that swallowing reflex that allows for food to just slide right through once it gets into that area. Okay? All right. So here's something, this is kind of a case study, but it's real life, actually. My daughter, when she was in first grade, she was complaining. She didn't even come to me. I could just hear her like whimpering in her room. And I went up and I was like, "What's wrong?" And she said, "My stomach hurts." And I said, "Well, what did you eat?" And she goes, "I ate money." And I go, "Oh." And so then I said, all I could think about was like, is this a penny or a dime, right? So I asked her, I said, "Alex, did you swallow a penny or a dime?" And she goes, "No, I swallowed the big one." So she swallowed a quarter. What was happening, her symptoms were this. She was clutching her chest and then relaxing. And then clutching her chest and then relaxing. And then she'd fall asleep. And this just went on over and over. So I finally took her to the doctor. And I have her x-ray with me. They knew I taught physiology. What she was exhibiting, and I'm going to talk about this in a minute, is what's called secondary peristalsis of the esophagus. So, this is what it looked like. I mean, you could literally take this a quarter out of your pocket. Can you guys see that? I mean, it was like stuck in her esophagus. And what was happening was, this is a coordination between circular smooth muscle and longitudinal smooth muscle. And what it was doing is it was called secondary peristalsis, trying to push that quarter into her stomach, right? Now, this is how it works. I think the ladies can understand this a little better than the guys, so I'm sorry about this. But have you guys ever lost the string in your sweatpants? You know what I'm talking about? The string in your sweatpants. So you have a knot at the end. This is how peristalsis in the esophagus works. To restring it, you usually have to pinch the back of the knot and kind of push it forward. And then you take the fabric of the sweatpants and you like push it, trying to get that knot around the sweatpants. That's peristalsis, right? The circular smooth muscle pinches the bolus of food, and then the longitudinal smooth muscle shortens the esophagus. It pushes, right? Pushes the food down the line. That's peristalsis. So that's what she was exhibiting, was a secondary peristalsis. She would clutch her chest when it was happening, and then relax and clutch her chest. So just to finish that story, this is what they had to do. They put her in a baby, literally a baby straitjacket. They had to put, make her hands go behind her back like this, and then they tubed her. They had to actually push the quarter into her stomach. And then they said, once it gets into the stomach, she'll pass it easily. This happens all the time. That's what they said. So they said, oh my gosh, you can't believe what kids will swallow. I mean, watch batteries or, Legos, all kinds of stuff gets stuck in their esophagus. So then it was my job, a day later, to actually filter through her poop, right? Because I had to make sure it got out. And it sure enough, it did, a few hours later. And I could report it to the doctor. But that's what happens. So that's what peristalsis is. Pretty good, right? Now you know a little bit more about how that works. All right. So let me finish up with this particular slide here on the right-hand side. See, I did make the esophagus a little bit more dramatic. All right. So this is the lower esophageal sphincter. Again, it has a basal tone associated with it, which is all like mediated through different neurons. So it's usually when you're not eating a meal constricted, and especially when you have food in your stomach, it's really constricted. And then it's relaxed by the vagus nerve. So it's all kind of mediated through these chemicals, signals, ATP through adenosine receptors, or what's called vasoactive intestinal peptide. We'll talk more about this on Friday, which helps to relax the lower esophageal sphincter. Okay? So there's two major pathophysiological states associated with the lower esophageal sphincter. One is esophagitis. Let's just start with that one. Whenever you hear the term itis, you can always think of inflammation, right? So this is an inflammation of the esophagus, esophagitis. This actually happens when there's an abnormal relaxation of the lower esophageal sphincter. What happens? You get acid that bubbles up from the stomach and starts to really affect those epithelial cells in the esophagus. You guys know what that is, right? Heartburn, right? That's heartburn. It's also called GERD, or G-E-R-D, right? That's gastroesophageal reflux disease, okay? So that causes esophagitis, which is usually an abnormal relaxation. Now, smokers, a lot of times, have an abnormal relaxation. Or women that are pregnant suffer from heartburn quite a bit because of an abnormal relaxation, okay? Now, what happens is if you let it go too long, then you can build up scar tissue in this area, and now the lower esophageal sphincter fails to relax. Okay, that's a big problem. I actually knew somebody that had esophagitis and then didn't do anything about it, and then built up all this scar tissue, and he literally had to go into the hospital to be tubed because he got food stuck in this particular area. Super painful, but that's called achalasia. Achalasia. So that's that's when the lower esophageal sphincter fails to relax. Okay? All right, so that's a good place to end, you guys. We're going to start with the stomach and small intestine on Friday. Have a good day, you guys. Yes. Oh, I did. And you're wanting to take it on the 9th or the 12th?
The 12th. The 9th is the day that I already have a bunch of classes.
Okay. So. Okay, so we're scheduled on the 9th.
Yeah.
But my other class is scheduled 10-10, Monday, Wednesday, Friday. Oh, it is Saturday the 13th.
Okay.
Can you do that on Saturday?
I think I could, yeah.
Okay. So that's 1:30 to 3:30, Saturday, May 13th.
Okay.
Does that sound okay?
That sounds good.
Yeah, thank you. They went through the Scantron today. If you wanted to send me an email, I could grade your diagram super quick. Okay. Yeah, Greg actually threw me the Scantron. So I should get that logged in. Yeah.
I just have a question with the posters. Is there supposed to be like one slide?
One big slide.
Okay,
Yeah.
so just one PowerPoint slide.
Yeah, and you can
Okay.
adjust the dimensions. It's in that instruction sheet.
Okay.
Adjust the dimensions so that it's just one big slide.
Okay, so I'm on the PowerPoint. So I downloaded one of the templates from that website, and then I just use one
Yeah.
PowerPoint page
Yeah.
slide. Okay.
Yeah, perfect.
Okay.
Yes, awesome. Yeah,
And then when you have it on Friday,
just open it up. You can send me, like, if you have it on Google Drive or something, you can just share it with me. Okay. That's probably the easiest way. And then then you wouldn't even have to turn it on in on me.
Right.
Yeah, I can just see it when it's all done.
okay,
Yeah.
and then are you going to be looking at them like Friday or
No.
Saturday?
I literally don't care if you're not even close to being
Sweet.
I just will like look at it and go, oh,
Okay.
yeah, I'm not giving you a score or anything. I'm just just making sure that you're starting. Okay, perfect. Yeah.
And then, yeah, we have to.
Oh, that's right. Okay, yeah, I will do that.
And then also I talked to you yesterday about
Yes, and you did. Okay.
um,
No. Okay. Let me just have a chance. It probably won't be until tomorrow. I have an administrative day. Okay. I have 2 more meetings today, so I probably won't get to it today, but I will get to it tomorrow. Yeah, that sounds good.
and thank you for marking the time.
Oh, yeah, good. And did you like you needed more copies?
We need more 5-hour energy.
Okay. All right. I'll get one.
Thank you.
I was gone Friday.
Okay, yeah. Yeah, definitely.
Hi. I just have a question. So you're going to Colorado?
Yeah.
Isn't there like, because there's this method that they use, it's like water, salt, and sugar,
Yeah.
or something that they like give to how to cure it in like third world countries.
That's really, yeah, I will be talking about it. It's called oral. Yeah, I will talk about it. That's a great question. And you'll see it in this. Okay, so we're going to talk about actual transport, like we did in Deglutition. So you're going to see the sodium glucose co-transporter. It's called oral rehydration therapy. Yeah. And you basically cannot give someone who's suffering from cholera just pure water. It'll go right through them. And the reason is, and this is interesting, we talk a lot about aquaporin channels in the gut, but there's not many aquaporin channels in the gut. But you saw how much water was reabsorbed, or absorbed. Yeah, not reabsorbed. So how is the water actually being absorbed? It's mainly thought now that it is being absorbed through the sodium glucose co-transporter.
And so then it's like a water bottle.
Yeah, not even water follows. It's actually attaching to the sodium glucose co-transporter. And that the water is actually moving